Today we will study about the topic molecular basis of cancer, the role of genetic alterations in causation of cancer. So what causes carcinogenesis? So non-lethal genetic changes lies at the heart of carcinogenesis, students. And why not lethal change? Because if the changes are lethal, the cell will die and not proliferate. So what is causing the initial damage? Initial damage is caused due to environmental exposure, germline mutation, or simply due to bad luck or random causes. So environmental exposure can be microbial, can be due to physical agents like UV radiation or chemicals. Microbes such as viruses can cause the initial damage. Germline mutations in sperm or ova, these are heritable changes that are transmitted from parents to offsprings. It is important for us to know what neoplasm exactly is before discussing the topic of carcinogenesis. So neoplasm is an abnormal mass of tissue, the growth of which exceeds and is uncoordinated with that of the normal tissues and persists in the same excessive manner even after cessation of the stimuli which evoke the change. So the neoplasm is an abnormal mass of tissue whose growth is more than that of the normal tissue, and the growth persists even after the removal of a stimulus. So how is a tumor formed? A tumor is formed by a single precursor cell which has incurred some genetic damage by clonal expansion. So single precursor cell form a clone of daughter cells, and since these daughter cells are cloned, they will be having the same set of mutations as the precursor cell. Since a normal cell is constantly exposed to stimulus such as harmful physical agents, chemical agents, or microbes, it is constantly exposed to carcinogenic stimulus. So why don't it changes to cancer cell normally? Because there have to be some protective mechanism within the cell, that is the normal regulatory genes are there for it, such as tumor suppressor genes, genes involved in DNA repair, genes which products are responsible for regularly repairing the DNA, and there are some genes that regulate apoptosis, that is the cell death pathway. And also there are some genes, proto-oncogenes. Proto-oncogenes produce normal proteins and they have normal functions. Let's start with the proto-oncogenes, students. So if there is a mutation of proto-oncogene, it will transform into an oncogene, and this oncogene is not producing normal protein, it is producing oncoprotein. So if there is a mutation of proto-oncogene, it transforms into an oncogene which produce oncoproteins, and mutations, they cause increase in one or the normal function of the normal genes, okay, and or may impart a new function. And since they are gaining a function, these mutations are gain of function mutation. They can transform the cells despite the presence of the normal copy of the same genes, thus they dominate over their normal counterparts. In this aspect, they differ from the tumor suppressor genes. How? There is a loss of function mutation and they behave recessively. Okay, so let's study about the tumor suppressor genes. By its name suggests that they are suppressing tumor. So let's see it how. The function of the tumor suppressor genes is to act as their products act as brake on the cell proliferation. How? They act as brake on the cell cycle pathway. Okay. So we know the function of cell cycle. The cell is dividing. The mitosis is happening. Mitosis or meiosis division is happening due to cell cycle. So cells proliferate via the cell cycle. And if excessive, can lead to formation of abnormal growth or say neoplasm. But here there are breaks due to tumor suppressor genes. But if there is a mutation if the function of the tumor suppressor gene is lost due to some region, due to some mutations like deletion, what will happen? This break on cell cycle will be released and the cell will keep on proliferating and form a mass, which the growth of which may persist even after the removal of a stimulus, which we call neoplasm. So they behave in recessive fashion, that is, most instances both alleles must be damaged before neoplastic transformation can occur. But there is an exception to this rule. We call it haploinsufficiency. So here loss of only a single tumor suppressor allele is required, and it reduces the activity of the protein which is encoded, and this is enough to release the brakes on cell proliferation and survival, and the cell will proliferate to give rise to neoplasm. Now let's study about the apoptosis regulating genes. There are two types of apoptosis regulating genes. Some genes have products which suppress the apoptosis or cell death pathway. So if something is suppressing apoptosis, it will lead to cell proliferation. Now there are some genes whose products promote apoptosis. If something is promoting apoptosis, it will not lead to cell proliferation. So if there is gain of function of the products which suppress apoptosis, more and more cell proliferation will occur and carcinogenesis can occur. If there is loss of function of products which promote apoptosis, what will happen? Again the cell proliferation will occur. So to understand this, you must know the definition of apoptosis. Apoptosis is a pathway of cell death that is induced by tightly regulated suicide program in which cells destined to die, they activate intrinsic enzymes that degrade the cell's own nuclear DNA and nuclear and cytoplasmic proteins. So what is happening inside the cells which is destined to die? There inside the cell, some enzymes are activated, some intrinsic enzymes are activated, and these enzymes are destroying the cell's DNA and proteins. Now the fourth regulatory genes are DNA repair genes. Since a normal cell is constantly exposed to harmful stimulus, genetic damage is continuously occurring. Okay, and this non-lethal genetic damage, if not repaired, keep on accumulating. So for that there are DNA repair genes whose products repair the genetic damage constantly. What will happen if there is a loss of function of this DNA repair genes? If there is loss of function mutation, the affected cell will continue to acquire mutation at an increased rate, and this state is called as mutator phenotype. And since there are numerous genetic damage which are keeping on accumulating, it will lead to instability of the genome. Now we will start stepwise acquisition of complementary mutations. So how is carcinogenesis happening? It results from accumulation of mutations in a stepwise fashion, and it happens over time and not suddenly. The phenotypic attributes of malignant neoplasm are referred to as cancer hallmarks such as excessive growth and local invasiveness. Since genotype is changing to that of the cancer, phenotype will also change. So there are mutations which contribute to development of malignant phenotype, and these mutations are called as driver mutations. So the first driver mutation is called as initiating mutation. First driver mutation is initiating mutation. But can a single mutation fully transform a normal cell to a cancer cell? Students, the answer to this is no. No single mutation can be fully transforming. So development of cancer requires additional driver mutations, and it takes considerable length of time for this to happen. And cells bearing initiating mutations may be found in blood samples taken as long as decade before the diagnosis. So this have gave rise to an idea that these cancer cells arise from the cancer stem cells, cells with stem cell-like properties, as they are capable of self-renewal and have long latency period. So let's see this slide, students. Here we can see in this diagram that a normal cell, if it is exposed to a carcinogen, it induces mutation in it, and an initiated precursor cell with stem cell-like properties is formed. This is what we studied in the slide. Now next what will happen, that there will be mutations affecting genomic integrity. We study about the loss of function mutation of DNA repair genes was affecting the genomic integrity, and a precursor with mutator phenotype was formed. We saw in that figure that there are certain genes which maintain genomic stability or integrity, and loss of function appear to be early step on road to malignancy, particularly for solid tumors. Also, this also increase greatly the frequency of passenger mutations, and these passenger mutations have no phenotypic consequences, students. Now next what will happen, the tumors will evolve genetically under the pressure of Darwinian selection. You know the Darwinian selection theory, students, the survival of fittest theory. So what happens, now the tumor cells will proliferate to give rise to a population of cells. So there are numerous cells within the tumor mass. So by the time tumor comes to attention of clinician, it has gone through minimum 30 doublets, and during this process there is competition among tumor cells for what? For nutrients and microenvironmental natures, and some subclones, they have the capacity to win this contest for nutrients, and they will dominate, and this tendency of tumor to become more aggressive over time is referred to as tumor progression. And so with time, what will happen? The constituent cells of tumor will become extremely heterogeneous, as some are more fit and some are less fit. You must have heard the story of the Galapagos Island, the fringes there, the survival of the fittest theory of the Darwin. This is analogous to that. So how do we know that genetically distinct subclones really exist in any particular cancer? With the help of techniques like DNA sequencing, mutations unique to a subset of tumor sites have been identified, and this second type of mutation can be used to create a tumor family tree, which is analogous to evolutionary tree for flinches on the Galapagos Island of Darwin, students. Let's revisit that summary diagram. So what's happening that a normal cell is exposed to carcinogen, and this will induce mutation, and an initiated precursor cell with stem cell like properties is formed. Then mutations affecting genomic integrity are acquired and precursor with mutated phenotype is formed. Now this will acquire additional driver mutation and a founding cancer cell will be formed, which will acquire additional mutations, and there will be emergence of subclones, and under Darwinian selection pressure, the cells will evolve to form genetically heterogeneous cancer. So that's all about this topic, students. Thank you.